Is everybody today? Hi. Sorry? Yes, this is just a continuation for our last lecture, and the one on cytokines that is already there. And I didn't quite have the chance to review the cytokines one, so it's basically exactly what you have there. Yes. Oh, thanks. I don't have time to do anything, just put some gel style. Yes, with the makeup exam, if you took it, it is what it is. I did not get the scores back yet. And if it is better than your other score, then I'll put the new score in. But no, I'm not going to curve it because, as you see, it was basically exactly the same test as the makeup too. And basically, that's going to be it. If it is better, I'll change the grade. And if it is not better, so you keep your curved grade for exam 2. So no panic. All right. Any more questions before we dive into Speedy Gonzalez mode? Yes. You can't see it. It's just the one with cytokines, and I have some slides that are just finishing up on the last lecture. Yes. Chapter 15. I think I did. I think I checked on it last night. But I'll check on it again just to make sure that the updated version is there. Okay. But in any case, you always have the videos, right? And I'm really making sure that I'm keeping up with uploading the videos in a timely manner. Otherwise, I get totally confused with the other class and which video was up, which video is not. It's a lot of videos to juggle. Okay. All right, so are we ready for cytokine? To finish the cell-mediated and go into cytokines? Sure. Are we cool? Yeah? This should be a fun and quick lecture. And I just realized that, of course, next week, I think, is our next test, is it? No? The week after. Oh, the week after. Oof, okay. Oh, I tell you, I can't write any more questions. I'm getting question traumatized. It's a lot of questions. Okay, so you do have a little more time to study, right? And also, keep on submitting those extra credit questions, because eventually, I think most likely after your final exam, the final day, that's the time that I will have to actually sit down and calculate all those, input all of those extra credits. But I am trying to keep up and either put, thumbs up or a little comment on the questions, as you submit them. So, okay, I couldn't resist. We're going to finish up the cellular immunity with the serial killer, right? I just thought it was a cute thing to put there, or kind of, I don't know, politically incorrect. Well, you know what I'm talking about. Gotta have a sense of humor. All right, so these guys are serial killers with an S, right? So what are we talking about here? Cytotoxic T cells, right? So those are your CD8 positive T cells. So, okay, guys, let's pay attention because there's a lot of information here, right? It's going to take a lot of concentration. Okay, so your serial killers are your CD8 positive T cells, and they have a very good job of killing their targets, right? And not only they have to kill the targets, but the killing has to be specific. You got to be directed specifically at the target cell. And of course, it has to be focused. They don't want to be killing any healthy cell that might be close to the target cell, right? And why is this important? Because remember that when fast-to-fast ligand killing doesn't work right away, and these cells have to use those toxic perforin and granzymes to punch holes on the cell membrane, if those cytokines, if those enzymes hit any cell that is close by, then the healthy cell might die too. And you don't want that to happen. That would be really bad collateral damage, right? So how is it that these cells accomplish such a focused and direct killing, you know? So what happens is that this T cell here, okay, so this guy here is the T cell, is the T cytotoxic cell, like right there. When it lands on the target cell, it makes a little suction cup at a spot on the membrane on the target cell. And this is what is shown kind of like in a 2D fashion over here. So this here you think is kind of a suction cup represented there. Now, when it feels, due to adherent molecules here, they're not showing, right, in this diagram, when it feels that it made a good suction cup here, then the contact of those adhering molecules send a signal to the T cell that is going to help to upregulate the calcium level in the cytoplasm, okay? So this T cell, after it gets upregulation of this calcium level, is going to allow docking of those granules that are filled up with those powerful punching enzymes, okay, is going to allow docking and fusion of the membrane and opening up of those granules only at the area of that little suction cup, okay, or isolated cup. It's not quite suction, it's kind of isolated, okay? So this is one method that allows for target killing. So now the cell gets there, punches holes, and once it knows that the holes are punched, then it can just, you know, go and kill somebody else. Okay? It doesn't have to be sitting there waiting for that cell to die. Okay. Now the perforin and granzymes, the ones that go here and make these little pores, they are doing basically the same kind of interaction as we had seen before with the innate immune system, right? Remember C9, right, of the complement system? So that's the one that also allows this. And you also had a hole punching function of C6, 7, and 8 that we didn't look into a lot of details, but they can also do the same type of job. Because remember that a person that is deficient on C9 of the complement system doesn't really have to worry much about immune deficiency because the other complement proteins pick up their function, right? So here is another, here are other molecules that can also do the kind of job on punching holes on target cells. Okay, so these serial killers, as I mentioned, is direct and focused, right? So here we know that these guys are your CD8 positive, and they are going to target those MHC class 1 molecules that are showing antigens that are strange, right? Now look at this, how interesting this is. So this T cell that's in there interacts with the CD1, right? And sends a signal to kill this cell. And now it has to go and find where are the other cells that are also, for example, infected with that same virus that is showing the same antigen on CD1. So it can't just sit there and watch the cell die. It has a job to do. It has to find all the other cells that are infected, okay? So it goes there, it sends a signal to kill, and then it flies away and goes to look for somebody else to kill, okay? So this is what it's saying here. This CTL is going to trigger apoptosis, and then it just doesn't sit around. It just flies off and look for its next victim, right? And forgets about the cell that it just hit because it knows that it's going to die, okay? So it's very effective in this way that it can kill several cells in a short period of time. Okay. And here we go with the story with the natural killer cells. So these two observations here, I just put them in, so I didn't upload. So I can upload this also, but you know, it's just a couple of observations that I put in the slides. Because the way the natural killer cells, so those are your NK cells, right? The way that your natural killer cells kill the cells, this is different than your cytotoxic T cells. So these guys are not going to look for those MHC1s, right? So what they're going to look for is absence of MHC1, right? And not only that, but this cell also has to balance out its activating signals against the deactivating signals, okay? So basically, as we have here from osmosis, it says that it follows a set of rules that is going to tell it either to kill or not to kill the cell that it interacted with, okay? So it has receptors on its surface, right? So 3 receptors that are important for you to remember, right? One of them is this guy here, NKG2D. So this guy here is an activator, is an activating receptor, okay? NKG2D is activating. If it finds molecules on the surface of the target cell that bind to it, then it is going to try to kill the cell, but not right away, okay? Because all of the cells, all of the target cells are going to have some ligand that is going to bind to this receptor. Now, in order to balance out and just not go and killing every cell that it interacts with, it has these other receptors here. One of them is NKG2A and KIR. So these 2 guys over here are inhibitory, okay? Those are inhibitory. KIR is kind of easy because it's a killer inhibitory receptor, so that actually makes sense. But natural killer G2A is kind of confusing because now we don't know if the A is inhibitor or if the D is inhibitor, because they kind of have the same names and 2 little letters to differentiate them, and that drives anybody crazy, right? So I don't know what you're going to associate with A being the killer. All right, so A is the one that is inhibitory, okay? And D, okay, D is D killer. I don't know. This guy is activating. This guy is inhibitory. Okay, so you have activating receptors, you have inhibitory receptors. Okay. So how is it that the cell is killing? If it finds MHC1 on a healthy cell, so this guy here is a healthy cell, right? Then it doesn't do anything because it's not supposed to, right? If it finds that this unhappy guy doesn't have MHC1 on the surface, now it's going to decide to kill it. Why? Because it can? No. Well, because cells that do not have MHC1, we mean nucleated cells that don't have MHC1, are indicative of either viral infection or transformation, meaning cancer cells. Okay? Cancer cells downregulate MHC1 because cancer cells have strange proteins that are being expressed, right? Because it's transformed, it's not your natural cell. If it puts a piece of a transformed protein on the MHC1, then natural killer cell is going to identify the cytotoxic T cell, right? TC cell is going to identify that as a threat and is going to kill the cell. The cancer cell doesn't want to die, so it downregulates MHC1 in order to save itself. Now, it doesn't really save itself because when natural killer cell gets there, it doesn't find MHC1, then that's a signal to kill the cell. Okay? All right, so no MHC1 is a signal for natural killer to kill it. Now, if the cell has MHC1, but this MHC1 is not self, meaning it's from somebody else, right? Then the natural killer cell has a signal to go and kill the cell. And why is this a problem? Transplants, right? This is a big problem in case of transplants because most of the time you want to match those MHC1s, but if you have one of them that is mismatched, now you can have a problem of natural killer cells attacking the transplanted organ, okay? So that we're going to see more of that later when you get into transplants, but that can be a problem. But this is a signal for natural killer to kill the target, foreign MHC1 presence. Okay. All right, and this guy is also unhappy. Okay. All right, so how does the cell figure out if it should kill or if it should not kill? So usually, if it has inhibitory signals, those inhibitory signals are going to override the activation signals. But it's not as simple as that. That happens if you have the same levels of inhibitory and activating molecules, okay? So if you have here, for example, a cell that recognizes like somebody else MHC, and at the same time recognizes a viral antigen. This thing, okay, recognizing MHC self, okay, self MHC in this case means don't kill, but then recognizes a viral antigen, it means kill, and then the natural killer cell is going to say, should I kill or should I not kill, right? Because natural killer cell is not going to identify that antigen on MHC1, right? It either identifies the presence or absence of MHC1. So it doesn't really look for that antigen. So in order to figure out if the cell should be killed or not, it has to measure how the other receptors behaved, okay? So if you have more signals that are activating, right, that are interacting with the activating receptors, than the signals that are activating with the inhibitory receptors, then the natural killer cell is going to kill the cell. If it has the same level of activating and inhibiting, then it's not going to kill the cell because the inhibiting signal is going to win. Okay? All right, so is that simple? Make sense? All right, cool. Okay. Also, before I go into this one, keep in mind that that signal of activating and inhibiting receptor is the mechanism by which the natural killer cells don't attack your red blood cells, right? Because remember, the red blood cells are anucleated and they don't have MHC1 on the surface, right? So the natural killer cell gets there and say, oh, no MHC1, should I kill you? But then it has those inhibitory receptor interactions that tell natural killer cell, okay, I don't have MHC1, but I'm your friend, don't kill me. So those are inhibitory signals that are going to keep the red blood cell alive. Okay, so natural killer has another way of killing that is called ADCC. We have seen this guy before, right? Antibody dependent cell-mediated cytotoxicity or antibody-dependent cytotoxicity, okay? How is this happening? Pay attention to this receptor right there. CD16 is a receptor on your natural killer cell that recognizes the constant region or the Fc portion of your IgG antibody, okay? Now, when is this important? When your antibody recognizes an antigen that might be presented here at MHC1 receptor, right? MHC1 molecule, not a receptor, so MHC1 molecule. So antibody can bind to that antigen. And what's the use of it? Well, antibody is not going to do anything when it's bound here to that antigen, right? But what it is doing, it is opsonizing that cell. It is tagging that cell for killing by natural killer cells. So here comes the natural killer cells with the CD16 receptor, that interacts with the Fc portion, and then this allows targeting of that cell for killing. Okay, so this is antibody-dependent type of killing. And it is important that this receptor here, this CD16, recognizes the Fc constant region, because that's the part that doesn't change when the antibody goes for further improvement, right? When the antibody does a better binding mutation of the hypervariable region, right? Hypermutation. Yeah.
Are there different antibodies for opsonizing different cells?
Yes.
Like IgG is specific for opsonizing or natural killer cells. While
Yes.
Like,
yes. And remember,
IgE would be for opsonizing mast cells.
For opsonizing the target of mast cells, because your mast cells will have receptors that recognize the constant region of the IgE. And you know, constant region of IgE is different than the constant region of IgG, because the constant region is really what determines the isotype of your antibody. Okay? So in this case, IgG is going to be more specific for viral types of infections. So if a cell is infected and puts out this viral antigen on MHC1, right? So now that antibody can go there and bind to that antigen, and is flagging out the cell, saying, you know, something is wrong here. You know, if it is a healthy cell, I'm not going to be bound to it. So now natural killer cell goes there and tries to kill it.
So because of opsonization, even though it can be specific for specific antibodies to specific, for calling specific cells, even though we don't usually talk about it, antibodies are still a key part in dealing with viral infections.
Oh yes, oh yes, antibodies are very much keys for dealing with that, because not only they can opsonize like this, but they can also neutralize the virus and directly opsonize the virus, right? Because this antigen here can be found in the virus itself, right? This is a piece of the virus that is shown in the cell, because the virus has to replicate inside of the cell. Right? So the antibodies really have a lot of functions. Remember those five basic functions of the antibodies, right? So this is very important to keep that in mind. And this is like one of those challenging things about immunology, because everything is so connected. And then you're dealing with the situation of what to focus on first, because everything seems to be going in this everlasting circle of interactions, and everybody interacts with everybody else, and you're like lost in this huge redundancy of stuff, right? But it's important that you have the basic ideas of what they're doing, because if any of those fail, you know that first you're going to have a backup system, because you have all this redundancy going on. So the person can still, of course, survive and kill off the infection or the pathogen. Okay. So here, questions to practice and to think about them. Can you describe this type of killing, right? And then what are the differences between the killings of cytotoxic T cells and natural killer cells, right? So you should be able to think about those differences and identify them. Mm-hmm. So this is straight from your book. Okay. So yeah, so inhibitory and activating signals. Let's talk about that. Here comes your first question, and of course I forgot to activate that thing. So let me just lower this a little. 1 minute and a half. All right, time is ticking. Natural killer cells distinguish their target cells primarily through what?
Delete.
Okay, people, we're talking about natural killer cells here.
Legit. Managed to encapsulate. That's only that one molecule in a real.
Okay, so what's your answer here? It is C. Yes, C. Your natural killer cell is going to balance out if it has enough activating versus inhibitory signaling, right? And is going to decide to kill the target based on those. Okay. So here, when you need to stop that, yes.
I know I kind of like dive in around the question I asked earlier. What antibodies are like opsonizing for phagocytes?
For phagocytes, you have IgG, and this is also another type, but it's not for phagocytes, right? So IgG is mainly to bring the phagocytes. So the phagocytic cells are going to have receptors that are also going to recognize the Fc region of that. And then the question should be, why isn't that a phagocytic cell goes in there and phagocytoses this cell? Well, the problem here is that the cell might not be alone. It might be part of a tissue that is infected. So if the phagocytic cell wants to eat up that cell, it would have to eat up the whole tissue. And that's not possible. It's bigger than the cell, right? So then that's like useless. Phagocytic cell gets in there, it's like, oh, I can't eat you. I'll just let somebody else take care of it. Because that has to do with intracellular pathogen, not really with something that is floating around extracellularly that it can easily digest. Yeah, or virus or bacteria. Okay. Yeah, and this is really cool, like the way that these antibodies are working, because they are they can potentially trigger all this type of reactions. And then the system has to figure out which one is going to work where. So it's really like neat. Yeah. Yeah, exactly. It could activate the complement system. Yeah. So that's like cool stuff, right? Okay. So here, after the pathogen is eliminated, so you must downregulate that reaction of the immune system. And this is where those T regulatory cells are going to jump into action. So how is it that the Tregs downregulate the immune response, right? So here, basically we know that they're going to inhibit all types of immune cells, meaning innate or adaptive, right? So all of them, they get downregulated by the natural killer cells. Now, how is it that they do? They have this molecule on their surface that is called CTLA-4, okay? So CTLA-4 is for cytotoxic T lymphocyte associated antigen 4. It's like a mouthful. You have to take a deep breath before you say the name of this molecule, right? Now, what CTLA-4, it is going to bind and inhibit the B7 on antigen presenting cells. Okay, so remember B7 is, what is it used for? Especially in the dendritic cells, what the dendritic cell is using that for? Co-activator of those naive T cells, right? Without the B7, the naive T cells are not going to further differentiate to subtypes, right? So if your dendritic cell doesn't have a lot of those B7 on the surface, so that's the first signal that is not supposed to go and activate more T cells. Okay, so this Treg cell is first going to inhibit the activator. Okay, so that's a good idea, because if it tries to inhibit the effector cell, and then you have the activator activating a lot of cells on the other side. It's like you have a hole in the boat and you're trying to put out the water, but the hole keeps on filling on the other side, so it's kind of like useless, right? So it goes straight for the cell that is activating those naive cells and tells it, kind of like, be quiet now, your job is done, relax, okay? Go and be dormant or die. Okay, so a very nice signal, right? So it stops those dendritic cells, okay? Now, besides that, they have a lot of these interleukin-2 receptors on their surface. Remember, interleukin-2 is that cytokine that is going to trigger proliferation of a lot of those lymphocytes, right? So if your T reg cell, okay, if your Treg cell has a lot of those receptors and binds to interleukin-2, then interleukin-2 is not going to be available to go and activate other cells to divide, right? So this is at the very root of let's stop all this proliferation of cells and activation of cells, right? Not only it has the interleukin-2 receptor, but it also has other receptors. They're called adenosine receptors. They're basically doing the same thing as gobbling up adenosine so that other cells do not get activated by that. But we do not go through the pathway of why adenosine is really important for that. So we are really going to focus here on the interleukin-3 receptors. Okay, so interleukin-2 receptors on the Treg, they are going to gobble up all of these activating cytokines, so they're not activating anymore. They're going to be, quiet. Okay, so here is your CTLA-4, and here is your IL-2 receptor. Okay. Now, your Treg is going to make cytokines that are anti-inflammatory, or cytokines that are going to quiet down this whole mess. Two cytokines that you have to remember that are anti-inflammatory. This is interleukin-10 and TGF-beta. Now, in my mind, for some reason, I always get confused between TGF-beta and TNF-alpha. I think just because they start with a T, I don't know, but in my head, I'm always confusing them. Until I finally came up with something that says TG, okay, don't put the I in there, TGIF. TGIF is like, oh, I'm going to relax, right? So TGIF is like, whew, time to quiet down. So that made me remember that TGF-beta is the anti-inflammatory that's quiet down type of cytokine, okay? But there's no I in there, okay? No I, it's just TGF, okay? So those are inhibitory ligands. Once the Treg makes those cytokines, those are going to interact with cells that are activated, and they're going to put them in an antisocial mode. Okay, so the cells, they get the cytokines and are like, I don't want to work anymore. I work too hard. It's time for TGIF. Right? It's time to relax. I don't want to talk to anybody. Just leave me alone. I was already stressed out, so I'm just going to be quiet and die. It's a very optimistic kind of thing to do, but it's going to stop that immune reaction. Okay, so here, clonal deletion is the next thing that will happen. After the cells get quiet down, they will die. Okay, so they're going to be self-sacrificed. Okay, so that is simple. Okay. And here you have this other mechanism that we had already seen before. So you have your Fas ligand that is present on your let me see here. Okay, sorry, this slide is a bit out of place. Yeah, this is why I was a bit confused here. Fas ligand interacts with the Fas receptor. Remember the trimerization of the Fas receptor? Yeah. So these are cytotoxic cells, but also the way to quiet down an immune response is that you have those cytotoxic cells and natural killer cells, okay, NK cells, that they have already the Fas ligand on their surface, because this is part of the mechanism that they can go and kill target cells. Remember, Fas interaction, right? Now, when the reaction is supposed to quiet down, when you want to stop that immune reaction, what is going to happen is that your target cells are not there anymore. They are dead. Okay, they already went through apoptosis. So now your cytotoxic T cells and your natural killer cells, they don't have a stimulus to keep on going and killing targets. Okay, they have the ligand, but they don't have anybody to interact with. So what they do is that they start to upregulate their own Fas receptor on their surface. Because you see this guy here, it's sad. It doesn't have anybody to interact with. Okay, so it's still sad, and it is upregulating the Fas receptor. And what is going to happen is that it already has the Fas ligand on the surface, and now it has the Fas receptor, and it thinks, oh, I can interact with myself. And what's going to happen? It's going to trigger apoptosis of itself. All right? It's so desperate for some kind of interaction, it interacts with itself and it kills itself. A kind of sad story. That's why it has a sad face. All right. So this is a way. Yes.
So is the Fas receptor native on the T cell, or is it the Fas ligand that's native?
The ligand is on the T cell, because the ligand is looking for receptors to activate on target cells. Remember that the receptor for the Fas, it has to be trimerized, right? Because you have a Fas all over the place on cells, but unless they get trimerized by this receptor, by this ligand, then the signal is not going to go inside of the cell for apoptosis. Okay?
Is the T cell the blue cell?
Okay, these are different types of cells. Okay, so this guy here is a cytotoxic T cell. This guy here would be a target cell. Okay, and now this guy here, yeah, I should have kept the color code. This guy here is a T cell, or is a natural killer cell that had a lot of Fas ligand. So let's put more Fas ligand over here, and not a lot of Fas receptor, but it starts to upregulate the Fas receptor because it has to bind to its own ligand and kill itself. Okay, yeah.
So, and to dampen the immune response at the end after the pathogen is already taken care of, the natural killer cells will go to dampen the response, but then your cytotoxic T cells and your NK cells will just naturally dampen themselves after a response?
Yeah.
Okay.
Yeah. If they don't have the stimulus of the pathogen presence, they have nothing to do.
So there's naturally going away.
Yeah, if they don't have constant stimulus to keep them working, they get bored to death. Yeah, and they kill themselves. They don't want. I mean, they want to retire, but they don't want to hang around while retired. You know. Okay. All right. So after you have all these cells that got downregulated, right? It was a clonal deletion. You have all this cell debris because you had a lot of apoptosis at the end of your autoimmune reaction. So you have to clean up the mess, right? So here we have this question: who is going to clean up all that dead evidence, all those dead cells? And one way that I thought about, cleaning up evidence of dead bodies is to call the mafia people. So these guys are your macrophages, right? The garbage eaters. Okay, so those are the macrophages. They are going to look for evidence that cells are dying or almost totally dead. Okay, so they are going to look for nearly dead bodies, right? So those are flipped bodies, like they kind of flip over, almost dead. What that means is that they are going to look for specific lipids that are externalized, and they're not supposed to be externalized. For example, in the classical one here is phosphatidylserine. I should have spelled it out, but the P is for phosphate. Okay, so phosphatidylserine is is a lipid that should be facing the cytoplasm if the cell is healthy, but when the cell starts to undergo apoptosis, the lipid is flipped to the outside of the cell, and that is a signal for your macrophage to interact with that externalized phosphatidylserine and eat up that cell. Here is kind of like the cell is shrinking, but basically this phagocytic cell is eating up that the thing that was a big cell over there. Okay. So this is how you clean up the mess. You use those macrophages. And you remember what kind of cell macrophage derived from? Monocytes, right, from the monocytes. All right, so now we get to this cytokine story. And I know it's a lot of learning objectives over here, but thank goodness we have been looking at cytokines throughout the lectures, like bits and pieces here and there, so you don't have a flood of annoying cytokines all at once. So you just have to remember where they are important and how they are acting, right? And very few new cytokines here. You just have to know when they are going to be acting and how they are really being useful. Okay. So basically this, when you talk about cytokines, you are talking about cell communication. I thought it was really cute to put cell phones in here, right? Not that type of cell communication. So yeah, cell phone. All right, so what are cytokines? They are soluble factors, and they're going to do cell-to-cell communication just because the cells like to communicate just like every other living thing, okay? They are going to also coordinate communication with microvesicles. The means of communication is not only cytokines, but you also have microvesicles and exosomes. There are even tinier types of vesicles. And mainly these parts of communication, they have to do with mainly eukaryotic cells. But even bacteria communicate with each other, okay? They have little speech bubbles that are types of exosomes that allow bacteria to communicate with each other. So anything that is alive will have a way to communicate, okay? So I put this guy here saying, now we're talking, okay? So basically cells cannot function in isolation. So anything that is alive will figure out a way to communicate with something else that is alive. Okay, so yeah, I like this video here that says even bacteria talk to each other, right? So if you're curious to know a little bit of how bacteria talk to each other, go and watch this video. And of course, I'm not going to test you on this, but this is a very cool example of how, for example, bioluminescence can be activated based on the density or the level of bacteria that are present at a certain space or within a host. Okay. So cell signaling, right? It's very critical for every type of function. So let me see what is important to know here. Okay. So mainly we're going to focus here on the cytokine type of signaling. We have seen several cell-to-cell contact type of signaling, like the Fas ligand and Fas receptor, or some other types of interaction, like the T cell going to MHC. Those are all cell-to-cell signaling type of work. But now, basically, this is non-direct contact by using these messengers. So we really focus here on the cytokines, but you can also have other types of messengers like lipid mediators or other things that not necessarily are cytokines or chemokines. Okay. So we have seen cell-to-cell interaction in case of leukocyte homing. So this is just a little review of what is happening. Yeah, we have seen all that. You can read on your own and do a little review. And here we get more on the function of cytokines, presence present basically everywhere in the immune response. So you're going to have cells that are in the adaptive and in the innate immune response that are going to be responding to the presence of cytokines, and they're going to produce those cytokines. Now, the same cytokine can be produced by different cell types, okay? And they're going to act on different cell types as well. Okay, and this we have seen before, the types of properties of cytokines for communication. So you have the cytokine that acts on itself, and that is the autocrine, right? You can give me an example of autocrine? Interleukin-2, that's a good example, right? But that one is not a cytokine. Is not released. Most of the time is not released.
It's not a cytokine, right?
It's not a cytokine. No, it's a ligand. It's not a cytokine. Yeah, it's not. Although there are some cancer cells that throw some Fas receptor or Fas ligand on the environment. But that's a different story. It's not a cytokine. Okay, now you have also the ones that are acting on nearby cells. Can you give me an example of a paracrine action? Interleukin-2. Yeah, this guy is awesome. So another example of autocrine and paracrine cytokine is interferon gamma. Okay, that's another one that you should remember.
You said both autocrine and paracrine?
Yes, yes. And then you have those endocrine ones that they have to jump into the circulation and hit some distant targets. Can you give me an example of that type of cytokine? I don't think I emphasized it much before, but I briefly mentioned it sometime. Those are chemokines, and they're acting locally, attracting cells to get there. Here I'm thinking more of inflammatory cytokines. Yes, interleukin-6, right? So there are 3 inflammatory cytokines that you always have to have in mind. They are interleukin-1, interleukin-6, and TNF-alpha, okay, tumor necrosis factor alpha. Okay, so these 3 are highly inflammatory, and they can jump into circulation and get to your brain and hit your hypothalamus and cause fever. Right? Those are examples of your endocrine. Okay. Oh, there they are. Okay. And this is what happens when I don't have time to review my slides. I was predicting what's in the next slide. Okay, so what else are these guys doing? Okay, this triad here. Not only they're going to the brain and causing fever, but they can also hit your liver. And they can bring up acute response. Let me see if I have it here. See, and liver. Okay, here, acute phase reactants. Okay, acute phase for acute inflammation, right? So those are your C-reactive protein, CRP, and also increase the production of complement proteins. Why would that be helpful? If you make more complement proteins, you are going to be able to attack pathogens more effectively, because remember, the liver is the one that is producing those complement proteins, including the mannose binding lectin, right, and other complement proteins, right? The liver is the one that produces those, and if you have an infection and you're using up your complement proteins, you're going to run out of it, right? You're going to run out of those proteins. So if you have an infection and there are cells producing those inflammatory triads of cytokines, now that is going to tell your liver that most likely you're going to need more of those complement proteins, because if it is a big infection, you're going to run out of them. They are not, abundant supply in the blood. It's a limited supply, right? But if you need more, the liver will produce more. Okay. All right, so there are 5 types of cytokines. So the first one here are the interleukins, and they are called interleukins because at first it thought, well, scientists or immunologists thought that they were only going to be used to for talk between the leukocyte, so interleukocytes. But then it was discovered that those interleukins are acting also on other types of cells. They're non-leukocytes. So, bless you. So interleukins is just like a general name for those cytokines. Second type is the tumor necrosis factors. So there's a whole family of them, right? So you have here tumor necrosis factor alpha and beta, and then you have this lymphotoxin or LT beta. And those are very important for a direct response on inflammatory response against pathogens and also against damaged or infected cells. And I don't think I have that here. So keep in mind that this TNF family, they can bind to targets and directly cause the death of the target cell. So directly, they're going to cause apoptosis of the target cell. Okay, so these are very important for this type of damaged cell killing. So here you're seeing this tumor necrosis factor that interacted with a tumor cell and causes apoptosis of the tumor cell. Or in this case, you would have several factors. They're interacting with several cells, right? It's a bit misleading, showing one cell interacting and the whole thing committing suicide. Kind of not like that. Okay, let me see. What else they do? They are going to cause cells in the blood vessels to upregulate those adhesion molecules. And remember, that's going to cause the rolling and attachment and extravasation of your immune cells to a site of infection, right? So if you upregulate those adhesion molecules, you're going to be calling the recruitment of those cells to that site where they are needed. So not only they increase adhesion molecules, but they're also going to increase the separation of those cells in the blood vessels or the cell permeability. So when the recruited cells get there and are interacting with the adhesion molecules, they're also going to have extra space here between the cells to do the extravasation and get to the site of where they are needed. Okay, third type here, there are your interferons. So you have 2 types of interferons. You have interferon 1, and this is your alpha and beta. Usually they go together, right? So alpha and beta. And those are very, very important for antiviral defense. Okay, so alpha, beta for antiviral defense. And then you have your type 2 interferon, which you have only one of them, and this is your interferon gamma. And that is very important for activation of those macrophages. Remember what it's doing for the macrophages? Increasing reactant oxygen species, right, ROS? Enhancing the phagocytosis and killing of those pathogens, right? And also is going to activate the natural killer cells. So that is going to be activating innate immunity and natural killer cell killing for elimination of whatever pathogen must be present. Okay. Besides those innate immune cells, you're also going to activate adaptive immune response. Like how? You're going to upregulate MHC expression, both of them, that is going to allow faster detection of antigens that can be present, either extracellular or intracellular. So interferon gamma is very good for that. Okay. So here interferon alpha beta, what it's going to do is that is going to mount up an immune defense. It doesn't do direct killing, okay? These are only cytokines. So they're going to send signals to nearby cells that are not infected. They're going to send signals for those cells to arm themselves up with antiviral proteins, because most likely they are going to get infected soon. So interferon alpha beta is going to basically put everybody on the lookout for a possible viral infection. So when the viral gets there, then the cell is already protected against that, the killing by that virus. Okay. Yeah, so this is, the infected cell is putting out those interferon alpha beta, and basically telling the other cells, save yourselves, I'm dying. Yes.
So is that from the great mRNA the cell or the surrounding cell?
The surrounding cells. These are effects that are going to be felt by the surrounding cells. Okay. So what's going to happen here? Interferon alpha beta is going to go in the extracellular space. Is going to find a receptor here on nearby cells, and is going to activate the intracellular part of that receptor to send a signal to the nucleus to upregulate antiviral genes. So then you're going to be able to upregulate genes that are going to chop up RNA, and you're going to increase protein synthesis and make more MHC, and do a whole thing that is going to allow the cell to not
So
just
increase protein synthesis?
yes is
Inhibit protein synthesis.
Okay, of the surrounding cell. Okay, this is misleading. This is of pathogen. Increase protein synthesis of the cell that is being protected. Okay, yeah, this is what happens when I borrow too many osmosis videos. Okay, what is it that's going to make sense here? This cytokine binds to a receptor. This receptor here is going to activate a cell signaling process that is called STAT1 signaling. Okay, so STAT1 signaling is going to send a transcription factor to the nucleus. That transcription factor is going to upregulate antiviral proteins. For you to get antiviral proteins, you're going to have to increase protein synthesis of the cells that have to be guarded. Okay? Now, some of those antiviral proteins are going to target machinery used by the virus that is going to decrease the viral replication, but that is too much stuff that we don't want to get into. Okay, so overall idea of what is happening here, you're going to tell your healthy cells to increase their ammunition to kill the virus when the virus gets there. And in order to do that, you have to increase protein synthesis of your cell that has to be protected. Okay. All right, so everybody's clear with that?
Yeah, that was really good.
Okay, great. Okay, so here, other type, you have here type 4, colony stimulating factor. They are also called CSFs, okay, colony stimulating factors. Now, these are quite important at the bone marrow and the thymus, because they are going to direct hematopoiesis and differentiation of your cells. Okay, so maybe in the back of your mind, if you can remember the B cell, the differentiation in the bone marrow, you see something that is CSF, something GM CSF, and CSF this and CSF that. So here you have an example of granulocyte macrophage colony stimulating factor, and that is like GM CSF, and that is going to tell this pluripotent stem cell that it should become, either like granulocyte or a macrophage, but it's only after exposure to these types of cytokines. Okay, so they're specific for, they're more important at primary lymphoid tissues. Okay. Your last type of cytokine is your transforming growth factor. and there are lots of them, and we are not going to go into a lot of details because that's enough, right? So your TGIF is the guy that gets everybody quiet, right? TGIF here, or TGF, beta, is inhibitory, right? It's going to quiet everybody down. It's going to bring down proliferation and differentiation, and is going to help your Treg cells, right? Is going to be also produced by the Treg cells. It's going to help Treg cells to quiet everybody down. Basically saying like, oh, the other guy didn't show up. Oh, well, I had a little thing that goes like, slow down. Everybody's going like, slow down. So that's the TGF, this TGF factor there. All right, so here, more specific things on how cytokines are working. So these are the properties of your cytokines. Okay, you're not going to have stored cytokine in a cell. They are going to be made as they are needed, and that is called de novo synthesis. Okay? The way that they are going to work is that they're going to look for a receptor. Okay? So they are going to find a receptor that they're going to interact with. Usually the receptors for cytokines, those are the big receptors. They are called the 7 transmembrane receptors or G protein coupled receptors. They have a lot of names. Mainly what this thing is doing is interacting on the outside of the cell. It doesn't go inside of the cell. Okay? It interacts outside of the cell and sends a signal to the inside of the cell. Okay? Without going inside of the cell. Okay, so interesting things about the cytokine. There is this idea of pleiotropism. Now, what in the world is that? One cytokine can do many things. Okay, is a jack of all trades. Not all trades, but several trades, right? So is one guy with several jobs. So it can exert different effects depending on the cell type that has that receptor. Okay, so what does that mean? For example, interleukin 1, we know that it can cause fever, but then it can go to the liver and cause increased expression of those inflammatory proteins or those acute response proteins. So depending on the cell type that has a receptor for it, of course the cell type is going to react in a different way. Okay, so pleiotropism. So here is, for example, interleukin 4 that can act on a B cell and cause activation and proliferation and differentiation. Now, if you have interleukin 4 present and interleukin 4 goes to a B cell, what is the type or the isotype of antibody that you're going to get out of that B cell? Ooh, time to get your brains to work. Ah, I can see the smoke coming out, almost. Think about it. Ah, okay. All right. Everybody's awake? Yeah, all right. This is you got to use a brain type of question, right? If you have interleukin 4 acting on a B cell, what is the type of antibody that the B cell is going to class switch?
There's only so many IGs I can say.
You already went to 3 of them and you only have 2 left. Is he? That was the last one. Oh my goodness. What is interleukin 4 going to differentiate Th0 cells towards? Ah, that's going to be on the test, huh? Th2. Why? Because you're going to produce, remember your dendritic cells? They're going to produce interleukin 4 and 5 when it found those parasites, those helmets. So you are going to need the Th2 cells, right? If you have Th2 cells and you have parasites around, if you're going to activate your B cells, you're going to need IgE around. You see how all this stuff gets connected, right? It gets really like this, and that, like pieces of the puzzle. And then you actually have to use your head to figure out what's going on, right? But I had mentioned before interleukin 4 and interleukin 5, right? And there is always a little partner that goes with those two, that is interleukin 13. Yeah.
You said Th2 is for the phagocytes?
Interleukin 2.
No, Th2.
Th2 is against those helminths, the worms, parasites.
But it's for the B cells or the phagocytes?
In this case, is the B.
Oh, shoot.
You see, that's the problem of redundancy or pleiotropism of those cytokines. You cannot say it's only important for this cell or the other cell. This guy is going to be important for, look at this, T cell differentiation into Th2 subtype. Thymocytes are not fully differentiated T cells, right? Interleukin-4 is also going to activate the mast cells. Why? This is going to act against the helminth, right? Because the mast cells, remember, has those receptors. They are going to interact with the Fc portion of IgE. So it needs to have IgE to interact with it. So then the B cell has to produce the IgE for this guy to interact with. Which other cell is going to be upregulated here? Eosinophils. Eosinophils. It starts with the E. Also has the receptor for IgE, right? So here we have one cytokine that is acting on many cells, and this is that property of pleiotropism. Do you have to know that? Yes. Okay. So let's rush here because I still have, oh, okay, not too many slides. I'll actually put my glasses to actually know how many slides. Okay, not so bad, good. All right, so in time, am I even finish on time today? Okay, 10 minutes, let's hurry. All right, redundancy. What is redundant? Yeah.
So with the pleiotropism, individual cytokine can do multiple things, what cells interact with, but they're released generally for one reason. Mainly, like for we know it was like for the helminth response, and then it affected like 500 cell type, correct?
Usually, yes.
Okay.
It's for one main type of infection.
Okay.
But it depends if it comes alone or if it goes together with somebody else.
Mm-hmm. But they're released for certain reasons, and then or affect just different cells that interact with.
Yeah, we're mainly going to focus on the cytokines that have one main job, and not really looking at interaction with other cytokines because that can be very confusing. Okay. All right. So redundancy. What does that mean? Different cytokines can have the same effect on a cell type. Okay. So if you don't have interleukin-4 around, interleukin-5 can get there and do the job. That's cool, because if you have a problem producing interleukin-4, you're not going to die out of an infection of a worm in your intestines, right? That means interleukin-5 will take over that job. So this is redundancy. If one soldier dies, another one will keep on going, okay? Here they have example of interleukin-19 and 20 on keratinocyte activation of increased expression of MAGE2, but I don't care if you know that. Just know what redundancy means, okay? Now, here, see these guys over here? This is what I meant. Interleukin 2, 4, and 5. Well, we know interleukin 2 is the guy of really important for a lot of cell types. But focus here on 4 and 5, okay? So these 4 and 5, they can go to the B cell, either one or the other, and they can tell this B cell to start producing IgE. If 4 is missing, 5 will take over the job of 4. Everybody cool on redundancy? All right. This is cool. Other properties here. Their actions can be added up together. They are also called this a synergistic type of action. Okay, so that means if 4 is acting alone, it has a little bit of a talk, but if 4 gets together with 5, now they can scream together, right? Their actions get louder. That's the synergy type. So 4 will give this a little push, 4 and 5 will give it a lot more push, okay? Synergistic effect. So that is when the cytokines are going together and acting to the same purpose. But if they are produced and they have opposite effects, they can have antagonistic effects, okay? Now, this happens mainly like at the end of an immune response when you really need to quiet down the effect of those pushing cytokines, okay? So for example here, If you have interleukin-4 being produced at the same time, then you're producing interferon gamma. Now, usually the negative stimulus is going to take over, and interleukin-4 is not going to be able to activate your B cell. Interferon gamma will do the activating. So they are antagonizing each other's action. Okay, so here we have blockage of action by interleukin-4 because the other cytokine will basically be sending a stronger signal. Okay, everybody cool with that? Synergistic, antagonistic. It's very intuitive, right? Okay. Now, cytokines are not hormones, okay? Cytokines are going to work at very, very low concentrations, like a little bit of it in your bloodstream, and you're going to have a big bang for it. For hormones, you need a lot more of their concentration in the blood for them to work. Okay. Now, let's see. Yeah, also, when you have stimulus of cytokine production, they can increase very quickly at a tissue. So this is like 1,000 times more of expression of the cytokine. And if you compare that to hormones, thank goodness hormones do not go through that jump, or you will be in deep trouble, right? Okay. All right. So we already looked at all that. You can just study that for at your own time. Okay. So here a bit more on cytokines versus hormone. Okay. All right. Let's see what is important to know here. Okay, so this, I have already talked of this, so you can just study that on your own. All right, so here a bit more on the action of interferon, which I already had mentioned before, but just to reiterate here, on your interferon type 1, which is alpha beta, and your interferon type 2, which is your interferon gamma, okay? And yeah, know how they are important. I just mentioned that. And of course, I did mention here importance of acute inflammatory cytokines. Then you absolutely have to know 1, 2, alpha, and interleukin 6. Interleukin 8 is also inflammatory, but I tend to forget about it. I don't know why. I guess it never comes to my head. Okay. Neglect the child. Okay. But you see, 1 and 6 and TNF alpha are really important for several of those functions. So make sure you go over this, and if you have questions, you ask me. Okay. Immune response. So here is just a little review of what we had seen before. Okay. Interleukin 4. Then you have here 2, 4, and 5. 4, 5, and 6. Okay. So we have seen that before. So just make sure that you understand what those are doing for the type of immune response that you need to have. Okay, now cytokines, let me see if I'll cover that now. Yeah, I still have like 5 minutes, so this is good. Okay, now cytokines can be a problem or can be helpful depending on disease state, right? So let me see what's going on here. All right, so when you have a problem of bacterial septic shock, first, when is it that something like this can happen? Septic shock. Have you heard of septic shock? Like, for example, when you have appendicitis, which is like quite common, and you don't go to the ER on time, right? And that appendicitis burst, then you're going to have septic shock because a lot of the bacteria that is in your intestine can, spread through your system, and now you're going to have a huge problem because the cells that come in contact with all those bacteria, they are going to signal, SOS state, and they're going to start producing a lot of inflammatory cytokines. Actually, they're going to produce it so much that we call it an overproduction of inflammatory cytokines. Yeah.
Has to do with, like, not so much the volume, but the rate at which the infection spreads and the rate at which the immune response happens. Like, it happens so fast and so big, that's what causes shock, right?
Yes, and now you're going to see the mechanism of how that happens, because bacteria is not supposed to be in your systemic space at all. So when you get it there, the cells that are producing these inflammatory cytokines are not only your immune cells, but also epithelial cells or other types of cells. Okay, so other types of cells can signal a problem and call on immune cells to go there. But now what's going to happen, look at this. If you have a lot of these inflammatory cytokines being produced in one space, then what's going to happen is that, first of all, how did you get a lot of these cytokines to be produced? Well, if you have a lot of bacterial components in that space, right, you are going to trigger a lot of cells to put all this dumping of these cytokines. What is the job of those cytokines? They are going to dilate the blood vessels, and they are going to one second. At the site of production, they're not just going to stay there. They are going to get systemically in the blood circulation. So they are going to dilate the blood vessel at the site of infection, but also they're going to get into the circulation, and because they are overproduced, they are going to dilate the blood vessels everywhere in the body. That is going to cause shock, because now the blood that is supposed to be pumped by your heart is not going to be in your core anymore. It's going to go all to the extremities, and your body is going to be depleted of blood. So this is what is called shock, and if it is caused by bacteria, you call it septic shock. Yes.
So when do the blood vessels constrict versus whether they dilate? Because I know at some immunological responses, they constrict the blood vessels to prevent bacteria from proliferating.
That I would have to look at specific types of situation. Like when it is a small situation, like for example, if you have infection on a cuticle, right, and you have edema, you have swelling. So that first you have increased blood permeability at the site for the white blood cells to get there. But then with this swelling, you are going to put pressure on the other vessels not to bring it too much, but it's a small and local type of effect. When you have systemic, what you're going to do is the bacteria is everywhere. So you really want to bring as many of the white blood cells to fight the bacteria as possible. And then this thing gets in the bloodstream and all your blood vessels are getting dilated. Okay. So this is why it's really important to, in case that happens, you have to be in a setting that can at first try to treat you very quickly with what kind of drug? What kind of drug is against vasodilation? Epinephrine. Then adrenaline, epinephrine. Oh, okay, I'm not going to test you on that. It's not for this class. Okay. All right, so we have a few. But depending on the extent of that problem, it might not be useful, right? Okay, so I'm going to stop here.